Good morning students, welcome to B.Sc-II Zoology class. So in the previous video we were discussing one of the morphogenetic movements that occur during the development of frog embryo from blastula. So we are talking about gastrulation in frog egg. What is gastrulation? Gastrulation is the process during which the blastula is converted into gastrule. During gastrulation, several processes are taking place. All processes together known as gastrulation. So there are four different morphogenetic movements occur during gastrulation. Okay, so the difference between blastula and gastrula is blastula has blastocele. Blastula has blastocele cavity, and this cavity is covered by single layer of blastomeres. Blastoderm is there. Now in case of gastrula, that blastoderm is converted into three germinal layers. Ectoderm, endoderm and mesoderm. Right? How the single layered, the blastoderm is converted into three layered, gastrula. That we are discussing. So during the gastrulation, four different morphogenetic movements occur. The first one is invagination and formation of blastopore. Second one is involution and closure of the blastopore. And third one is epiboly. And fourth one is rotation. So these are the four different types of morphogenetic movements occur during the conversion of blastula into gastrula. So the first morphogenetic movement, invagination and formation of blastopore, is completed. See, we already discussed how the invagination and blastopore formation occurs. Invagination is inward movement of superficial, blastomeres to form a groove or depression. That depression enlarges to form what? Another cavity, new cavity called archenteron. The opening of the archenteron to the exterior is called blastopore. The dorsal part of the blastopore is called dorsal lip of the blastopore. See how depression is formed at one surface, at the dorsal surface of the embryo of the blastula, due to the inward movement of the cells. Inward movement of the cells present at the border between Drake crescent and vestal hemisphere. As a result, these cells are pushed inward to form, they are forming bottle-like cells, and their narrow neck is connected to the surface of the blastula. Okay, now this depression is deepening because of what? Because of inward movement of superficial cells that are present at the surface. They are moving inward, and while they are moving inward, they roll over the rim of blastopore. Look here. So these diagrams show you the surface views of the gastrula, at the blastopore region. This is blastopore. And this part is called yolk plug. Means these are having yolky cells. So they are macromeres. They form by macromeres. Okay. So this forms yolk plug, and these are the diagrams to show you the internal changes that are occurring during gastrulation. Diagrammatic median section. Diagrammatic median section. So the first diagram, these two diagrams show you what process, what events occur during invagination. During invagination. Now look here. Very small depression is formed here. This is called blastopore. Blastopore due to the inward movement of the cells that are present at the surface. Clear? Now what happens? Look here. So here at the blastopore region, dorsal cells are moving inside. Initially, the dorsal surface is more active in proliferation of cells. And these cells are moving inward. As a result, pore is formed here. Very small pore is formed. Okay, next, involution. Involution means rolling of the surface cells into the interior of the embryo, rolling over the rim of the blastopore. Now the cells are moving inside. They are rolling. Here this part is having cells. These cells are, more active in proliferation, in multiplication. So multiply cells, what they do? They enter gradually. Suppose this is blastopore. This is the dorsal surface. This is dorsal lip of the blastopore. This is ventral lip of the blastopore. Now this part is very active. Cells are moving downward and rolling over the rim of the blastopore, and they come to lie just below the external layer. Here, like this. Okay. Now they are coming here, going inside. As a result, the organ size. This is what organ. Newly formed cavity, which is having opening to the exterior through blastopore. This organ size is also increasing. And these the cells which are rolling inside, they lie just below the external layer. Look here, the surface cells present at the dorsal side of the embryo are identified as presumptive. These cells are called as presumptive nervous system forming cells. Nervous system forming cells are present here. These are presumptive notochordal cells. Presumptive means in future these cells are going to be formed into notochord. In front of the presumptive notochordal cells, presumptive caudal plate, presumptive caudal plate. Cord refers to notochord. Before the notochord, some cells are there, and these cells are going to be formed into mesoderm. Okay, so these are entering. Look here, they have entered here. These cells are entered inside, and which cells are entering? Presumptive notochordal cells are entering. These are presumptive notochordal cells. So these are endodermal cells, presumptive endodermal cells. These cells going to be formed into endoderm. There are three germinal layers: ectoderm, endoderm, mesoderm. Okay. So these cells are entering, they are transformed into notochord, and these are going to be developed into endoderm. This is what? This cavity is what? This is blastocele. What is this? Blastocele. Okay. So thus, because of the what? Involution. Rolling of cells interior to the embryo. What is happening? Size of the blastocele is reduced. It is being reduced, and size of the organ is increasing in size. So at the surface of gastrula, at the dorsal region, dorsal column region. This part is increasing in size. It is increasing in size because of what? Because of the movement of dorsal cells and lateral cells. This is called dorsal blastocore. This is dorsal blastocore. And now what is forming? Lateral lip. Lateral lips are also forming. Lateral lips are also forming due to the lateral cells movement inside the embryo. Now the size of the blastocore is increased. It is increasing. First, only line like small longitudinal line like slit line structure is appear. It is increasing size around the yolk plug, forming half moon shape. And now it is forming complete circle around the yolk plug. And look here, the size of the blastocore is also decreasing what? Decreasing because of the contraction of all cells coming from all cells, dorsal side, ventral side, lateral side. They are all going inside. So they are going inside. These are all going inside. And this is what blastocele. This is what orchentron. Orchentron side. See, orchentron size is increasing. Blastocele size is decreasing here. Can you see it? So this is called involution. Involution, rolling up of surface cells into the embryo to form what? To form another layer of cells just beneath the external layer, outer layer. Alright. See, this is external layer, this one is internal layer. So now two layers are formed, two cellular layers are formed. This is external, this one is internal. Right? Internal. This layer is going to be formed into notochord. This part is going to be developed into mesoderm. This part is going to be developed into nervous system. Okay? So these cells are epidermal cells. That is epidermal cells. So as these cells are moving inside, these cells are also replicating at a faster rate, and they cover the entire embryo from all sides. So the entire embryo is externally covered by ectoderm or epidermal cells. Clear? So now looking. This is called epiboly. Epiboly means what? Epiboly means the spreading of epidermal cells over the entire surface of the embryo. So that all the vesicle side cells have entered into the cavity, into the embryo. See, all the yolk cells have entered. Inside. They have entered inside. Can you see it? So this is going to be developed into wall of the orchentron. Orchentron is nothing but presumptive gut, presumptive digestive system of the embryo. Digestive system is going to be developed by these cells, endoderm forms. Thus, so how many layers are formed here? See, this side also, mesodermal cells are present. They also enter here. Okay, so ectoderm, endoderm, mesoderm. So thus, at the end of gastrulation, single layered blastula transformed into three layered gastrula. Three germinal layers are very, very important for the further growth of different organ systems in adult form. Next, rotation. rotation occurs here. Rotation of the embryo inside the Wittlein membrane. Still, the embryo is externally covered by Wittlein membrane. Wittlein membrane is there. Wittlein membrane is nothing but fertilization membrane. Between fertilization membrane and the embryo proper, there is a cavity. That cavity is called as peri-Wittlein space. In that peri-Wittlein space, fluid is present. Because of the fluid, the embryo rotates inside the Wittlein membrane. So it rotates. Look here, I have shown you. This is the anterior end of the embryo, posterior end. Anterior end, posterior end. Anterior end, posterior end. It is rotating. Can you see it? Now, what has happened? This is anterior, this is posterior. So at the beginning, the blastopore is towards the posterior end. Now, at the end, the blastopore. This is what? Blastopore. Very small opening is there, in which the organ now opens to the exterior. This is blastopore. Blastopore comes to lie on the ventral surface, not towards the posterior end. This is posterior end, this is anterior end. Okay? So animal is, the embryo is rotated. It is rotated. Okay? So, vestral region of the initial embryo come to lie on the ventral surface of the embryo. Okay? Ventral surface of the embryo. So this is how the animal gets rotated. The embryo gets rotated within the Wittlein membrane. Thus, the single-layered blastula is converted into three-layered. Overall is three-layered embryo called gastrula. Here, ectoderm is the entire external surface is covered by ectoderm. Ectoderm is complete. Endoderm. So only the roof of the archenron is not completed, not having the endoderm. Okay? So mesodermal sheath is developed. Okay? So thus, three-layered embryo is developed, and that three-layered embryo is called as gastrula. This is how gastrulation completes in 4 morphogenetic movements. What is morphogenetic movement? Morphogenetic movement is nothing but movement of cells from one region to another region for the formation of germinal layers during the conversion of blastula into gastrula. So this is how the gastrulation takes place in frog embryo, frog development. Okay, thank you.